Priya Menon, and today we are discussing a new drug called Melflufen, which has been granted priority review by the FDA in combination with dexamethasone for use in patients with multiple myeloma. And we have with us a very distinguished myeloma specialist, Dr. Paul Richardson. Dr. Richardson is R.J. Corman Professor of Medicine at Harvard Medical School, the attending physician in the Division of Hematologic Oncology and the Multiple Myeloma and Bone Marrow Transplant Service at the Dana Farber Cancer Institute. He is a clinical program leader and director of clinical research for Jerome Lipper Multiple Myeloma Center at the Dana Farber Cancer Institute. Welcome to Cure Talks, Dr. Richardson.
Priya, it's a pleasure to be with you, and it's wonderful to be with Yelak and Gary and Cynthia.
Yeah. So as Dr. Richardson mentioned, the myeloma panel today consisting of advocates and survivors, your very own Gary Peterson, Cynthia Schmilowiski, and Yelak Peru. So everybody, thanks for joining in today. To all those people who are watching us live, we will be taking in questions at the end of the discussion based on availability of time. Please add your questions to the comment section of the page, or you can also email them to me at priya@trilix.com. Dr. Richardson, let's just get started with the discussion. And my first question, which I've been asking consistently on Cure Talks this season, is what everybody wants to hear on COVID vaccines. So what are you telling your myeloma patients about the vaccine? Should they take it, considering the fact that they're already immunocompromised?
Oh, no, we're all recommending that vaccination be a top priority for our patients. There are some broad guidelines that have come both from the IMF and the IMS, actually. The IMF guidelines are basically driven by the IMS, which is the International Myeloma Society guidelines. So I think when you look at these guidelines, you have to realize they're generated for US patients from an international perspective. So that reflects the fact that there are different vaccine platforms in different countries. The very good news for the United States is that we have the RNA-based vaccines in the form of the Pfizer platform and the Moderna platform. And to my mind, these are an extraordinarily impressive technologies where I think we're seeing vaccination protection rates in the excess of 90% in normal volunteers. So that being the case in our own patient population, we have to recognize that's probably going to be a bit lower. To give people a frame of reference, if you have an influenza vaccination as a ordinary person, your coverage is around 50–60%. In myeloma patients, we know that's a little bit lower. Probably the same applies to Moderna and to Pfizer, but nonetheless, and we don't absolutely know, but it's probably going to be still a remarkable degree of protection. So we're encouraging all our patients to vaccinate, but at the same time, once they're vaccinated, still not to let their guard down. You still have to be sensible to distance, to mask, and follow all the precautions. Some of my patients have been checking themselves for antibodies after they've been vaccinated. And I think we really just don't know how to interpret that information. So I'd caution patients about getting depressed if they then have an antibody test and it doesn't get the requisite number. I think we just don't know what that exactly means. What we do know is that the RNA platform generates not only a humoral response, Priya, but it also generates an innate response. And that's the beauty of the RNA platforms, because essentially it's not just your antibody response to the virus, it's the other parts of your immune system that are very important as well. Now, the question then becomes is, what about your treatment? Well, I think the IMS and IMF guidelines are very good, but I think one just has to encourage each patient to talk to their provider for the following reasons. Whilst we know that avoiding exposure close to steroids makes sense, really when it comes to drugs like immunomodulators, the concern might be not so much that it would suppress response to the vaccine, but might magnify side effects. So being on an IMiD, we know from studies we've done with other vaccines, actually enhances your response to the vaccine. The only caution would be that you don't want to actually magnify it to the extent that you have a side effect. So our advice has generally been, if you're on 3 weeks on and 1 week off of your immunomodulatory drug, be it lenalidomide or pomalidomide, perhaps having it on your week off makes the most sense. Avoiding being too close to a steroid. And again, it's practicality, not perfection. You know, if you've gotten a steroid a couple of weeks before and you get your slot for your vaccine, don't waste that slot. Get the vaccine. It's far better to do that than be, puritanical and say, oh, I got my steroid just a few days ago, I can't possibly do this. Remember, the vaccine effect takes about a week to be on board. One area that has been a source of confusion And I would say the same applies to proteasome inhibition. We have no evidence that proteasome inhibition actually blunts the ability to mount a vaccine response. So I wouldn't get too concerned about when you've had your bortezomib or your carfilzomib therapy. One area to be a little careful of is antibodies, because obviously when you get daratumumab, when you get isatuximab, elotuzumab, belantamab, mafodotin, whichever of the antibodies you receive, you probably get a little steroid with it. So perhaps being a little bit away from the antibody makes sense, and that I think is reasonable. So our common sense rule has been a week or two, or at least 2 weeks if you can. The other way we've approached this is to look at things like IVIG. Now, if you're giving an attenuated virus to a patient as your vaccine platform, that would be, for example, AstraZeneca, you have to be careful about IVIG because it blocks an FC receptor, which allows the immune system to grab things and learn from them. So if you're getting an AstraZeneca vaccine, that may not be a smart idea to be too close to your IVIG. Interestingly, for the RNA platforms, the same immunological mechanism does not apply. So whilst we're encouraging people to be away from their IVIG, a couple of weeks perhaps, it's not an absolute rule. And I'm a little bit concerned because some patients have been saying, I'm going to stop my IVIG for 3 months whilst I get vaccinated. Probably not the best idea, just simply common sense and working with your providers. Now, one class of drug to definitely be away from when you get your vaccine is chemotherapy. We know that alkylator-based therapy is potentially a blunter of immune response. And so therefore being as far away from chemotherapy as you can probably does make sense. And we know that from studies done, for example, with flu vaccines, where when flu vaccines and their responses were tested, of all the drug classes that were the most suppressive to vaccine response, interestingly, it wasn't actually even steroids, IMiDs, proteasome inhibitors. There weren't a lot of antibody-exposed patients in this study, but interestingly, it was the chemotherapy, the alkylators, that were the most immune suppressive. So a lot to digest for the audience there, but I hope those guidelines are helpful.
Definitely. So talk to your provider and get your vaccine. I think that's the message that is loud and clear here.
And don't be afraid. Don't be afraid of, if you've had treatment, don't be afraid of, I can't have my treatment because I'm going to be vaccinated or vice versa. Yep.
Yeah, thank you. That's very helpful, Dr. Richardson. Now, although we're still dealing with the complexities of the coronavirus, I believe myeloma progress is still happening, and we have a new drug which has been granted priority review. Dr. Richardson, what is Melflufen and why is it so exciting? And what do you predict will be the change in the myeloma treatment scenario with the addition of this new drug to the already existing treatment protocols, the myeloma armamentarium?
Well, Priya, I think the excitement around Melflufen is built on the fact that it's adapting a very key platform that we've just alluding to, alkylator therapy, into a very new and novel targeted approach that provides greater efficacy, which reflects not only greater activity, but also better tolerability. And so Melflufen is what we call a peptide-drug conjugate. It's a very clever way of delivering the alkylator warhead to where it needs to be. So to help patients understand it, if you think of a very exciting antibody like belantamab mafodotin, the antibody is belantamab. The warhead is mafodotin. That's belamaf or Blenrep for people who may know it under a different name. So it's a similar principle in a sense, except you're not using an antibody, you're using a delivery system, flufenamide, that allows you to get the warhead where it needs to be. So how does this happen? Well, basically, Melflufen, which has got the chemical name melphalan flufenamide, but it would be a mistake to think of it as, isn't this just glorified melphalan? No, absolutely not. It's very different. It's a lipophilic drug. And if you may think of your bone marrow, it's packed with fat. So when you need to get to your myeloma, you need drugs that are like to go into fat. Interesting enough, melphalan is lipophobic. So it immediately tells you is quite different. And second of all, when it gets into the bone marrow environment, it's only turned on by things called aminopeptidases. So what happens is this peptide-drug conjugate drops into the bone marrow. It's lipophilic, so it likes to go there. And then this key enzyme called an aminopeptidase, which is markedly overexpressed in myeloma, when the chemical drug conjugate enters the cell, the aminopeptidase cleaves it, the warhead shoots to where it's supposed to be, which is the nucleus of the myeloma cell. And because aminopeptidases are not expressed to anything like the same degree in normal tissues, you have literally a targeted delivery of your warhead. And what happens is that the warhead is trapped in the myeloma. So, if you go through a normal cell, the peptide-drug conjugate just scoots through. And if you go into the tumor cell, bloop, stops, warhead delivered. And there we are. So that's the rationale behind it. And preclinically, I want to especially acknowledge my colleague, Dr. Dharminder Chauhan, who really demonstrated very elegantly in preclinical models of myeloma, both on the bench and in our rodent models, that this was a real effect. So when he compared the behavior of tumor cells exposed to chemotherapeutics, completely different to when exposed to melflufen, with much more effects in melflufen. And the same thing was seen when he compared it to other drugs that we typically use. The very exciting thing is that he also showed that there were anti-angiogenic effects and all sorts of things that make melflufen very interesting. You may say, what about the anti-angiogenic piece? Well, when we give melflufen to patients, it works in what we call extramodular disease, which are big lumps of tumor. Those are very angiogenesis dependent. In other words, they depend on these new blood vessels to feed them. So we think there are differences in the way melflufen works that contribute to actually its efficacy. And one very interesting aspect of its biology is its ability to target high-risk disease. It targets the sort of stemness of myeloma, and it targets in particular, in elegant preclinical work, which has been demonstrated, 17p deleted disease. So there are effects that make it attractive as a drug beyond just traditional chemotherapy. The other thing from a patient perspective, which is key, is the tolerability. You don't lose your hair, you don't get mouth sores, and whilst it does cause suppression of blood counts, and we're very careful about that, and we're also investigating very carefully the potential risks of some of the side effects we see with other chemotherapies, nonetheless, generally speaking, it appears to be well tolerated and manageable. And we don't see the same rates of infections that we see with other drugs and other drug classes. And we similarly, because it's a very simple infusion once a month, it has the real attractiveness of not requiring too many visits to the clinic. You just come once a month, that's it. There are no other, there's no cardiac issue, no ocular issue, there's no neuropathy. We are very careful about blood counts, so that's one thing we have to watch, and the impact of that. But beyond that, it's generally well tolerated.
Thank you, Dr. Richardson. Now I'm going to invite the panel. Or Gary, you can start with your questions.
Thank you. And I first of all would like to thank you, Dr. Richardson, for all you do and you for putting this whole thing together, Priya, for all the work that you do and your leadership in the development of new classes of drug, and in also, new treatment paradigms. For example, I know that you were integral in coming up with the state-of-the-art care called VRD, or Velcade Revlimid Dex. That was, you did a lot of work on that. Also recently, selinexor was another one which you and a number of people worked very, very hard against a lot of, I'd say, ignorance on the part of the FDA because they didn't understand how to deal with the side effects. And the same thing goes true with this one. It's melflufen. It's a brand new class of drug. And in the last 16 years, we've had 10 new drugs and 10 new classes of drugs, all of which had dual designations by the FDA, meaning it was a priority review, a fast track, and as well as being an orphan drug designation. So I just wanted to thank you for all that you do for that. Right now, we have 7 new drugs, which we've talked about, and 4 of them were CAR-T, one was Indicel, or BB2121, and 3 others that we did. Another was CLR131, which is very similar to what you're talking about, but they use lipid rafts, but it's a whole new delivery process. And just like melflufen is a whole new delivery process, which seems to be irrespective of other, if you don't have to have BCMA, you don't have to have CD38, you don't have to have all these things. You have a whole different mechanism for targeting, which I think is fantastic. So Dr. Richardson, some people confuse melflufen for melphalan, but that is much like saying this highly targeted approach is the same as using a shotgun.
Yeah.
Meaning that,
that's very
so
good.
can you please explain why this is, so different and has so few off-target issues than, say, melphalan or cytoxin or anything like that?
But these are great questions. Gary, first and foremost, thank you for your very kind words. But I want to especially acknowledge that it's a real team effort, and it's been my privilege to work with a great group of clinical investigators, laboratory scientists, and actually it's wonderful to have you and Yelek on the call, and Cynthia too, because we've had great patient advocacy and participation in our trials, which has led to this incredible team effort. I do want to qualify one thing you said, though, just to be very clear. FDA have always, and we are very lucky we work with the myeloma team at the FDA. They're outstanding, and it's been a privilege to work with them. I think sometimes it's more difficult to deal with, when you're in very tough study populations of very resistant disease, where you're maximizing your therapy because that's the only way you're going to get a response. The side effect profiles are magnified. So FDA have a really important job of addressing safety, and I've found them incredibly collaborative in that role. But I do agree with you, outside of the FDA with selinexor, there was some uninformed comments being made by people who hadn't used the drug a lot to sort of say it was far too toxic. It's a challenging drug, selinexor, but especially when you combine it and use it just once a week, the results we're seeing are remarkable. So appreciate your comments about that.
I too like the FDA. I mean, they've given us, 16 or so drugs where there's only been 500 drugs for all 7,000 orphan diseases, and we got more than our fair share. So it's a miracle.
Yeah, I agree, Gary, and I think that's you hit the nail on the head. And I think, as to sort of be very clear, we've got the drugs you've mentioned are coming. We have 12 actual novel drugs approved since 2003. So you're absolutely right. We're in a very, very sweet spot. There are many more combinations approved, just to explain to the audience. So that's why there are approximately 30 approvals, but there are actually 12 drugs currently approved. Melflufen would be number 13, Idacel, hopefully number 14, venetoclax and others all coming, plus all those that you kindly mentioned. So I think an incredible team effort. But I also want to bring you back to a point you made about RVD, because this was where we brought drugs together, informed by the laboratory, built the platform. But what was very important about RVD is that we not only saw synergy in terms of its effect, but very importantly, we saw excellent tolerability. And for example, when we tried to look at, say, KRD versus RVD, frankly, I was expecting KRD to knock the socks off RVD. It actually didn't, because guess what? The side effects mattered. So for that reason, we really think sort of broadly about how to manage things. So that's why when we come back to Melflufen, I want to emphasize it really isn't just, as I say, glorified melphalan. It really isn't. We've deliberately sought to exploit a therapeutic index. By being more targeted, we can deliver that much more of the warhead to the tumor and minimize the surrounding damage to normal tissue. It sounds very simple, but it's actually very, very complex in what that actually results in. And what we've been able to show is that this delivery of the peptide-drug conjugate and the trapping of the cytotoxic moiety within the cell, very similar in the sense to belantamab mafodotin in a different context, that's Blenrep. This results in a very different pattern of killing in the myeloma. And as a result of that, in the clinical trials we've done, we've been very struck that Melflufen as a single agent, just simply combined with dexamethasone once a week, has generated about a third response rate, about a 30% response rate in patients in whom all other treatments have failed them. And very importantly, in those patients with extramedullary disease, which is such a challenge, we are seeing responses of the order of around 25% just with the drug alone, in a setting where really the best we see from other agents is much less than that. So with that in mind, the future of Melflufen, frankly, in my view, is going to be in combination. And there's some very nice data to share with you, Gary, where we combined Melflufen with daratumumab, we combined Melflufen with bortezomib, and again, always with the view of being practical, simple, also frankly cost-effective. That's where Melflufen Velcade is a good platform. We can actually engender great responses. And it's fair to say there's around an 80% response rate when you combine Melflufen with daratumumab, and a little bit less, but comparable with melflufen Velcade, even if you've had prior Velcade. So for that reason, we're very excited by the promise of melflufen.
Well, you just happened to just jump all over my second and third question.
Okay.
So I thank you very much. You just ran through them all in a very short time. Saved me a lot of talking. So with that, let's go to Cindy and see what she has to say. Cindy, you online? Cindy, we didn't lose you, did we?
No, I was on mute, so I have to unmute. You know, we always do that once during one of
Now
these
that's
meetings.
difficult to do.
Yeah. So Dr. Richardson provided so much information. He almost took all my questions too. But I guess I'm kind of curious about this peptide drug conjugate as opposed to an antibody. What's the difference? Like, what are peptides and why choose one? And, you know, with the antibodies, you either you target at CD38 or BCMA and things like that. With peptides, do you target them or like?
Yeah. So, Cindy, so it's a very good question, and Gary touched on it. Obviously, we're very lucky that we have specifically targeted approaches like CD38 and BCMA. CD38 is a marker expressed on all myeloma cells. BCMA, B-cell maturation antigen, is also expressed on myeloma cells, but they reflect part of a line of development in the myeloma ontogeny. Yeah, so you remember in myeloma, it's not just one line, it's an incredibly heterogeneous, multi-clonal, but clonal neoplasm. So I think one of the advantages potentially of melflufen is that it targets the tumor broadly. Aminopeptidases are expressed throughout myeloma, whether it expresses BCMA or it has less expression, whether it's CD38 overexpressing or for any reason its CD38 expression is downregulated because of prior treatment with CD38 antibody. The reality is that melflufen, in an indiscriminate fashion, if you will, targets the disease. And I would argue that this is the advantage potentially of the drug because it offers an ability to target what I call stemness. Now, stemness isn't just one cell. The complexity of myeloma biology is such that stemness is a composite. But this is, I think, a very important target. And we've known from the benefits that melphalan has delivered that targeting stemness is important. The challenge for melphalan is that, you know, obviously it has a side effect profile and a drug delivery challenge because we have to dose escalate it to get it to what we need to do, that makes it more difficult in the long term. In the short to medium term, obviously, melphalan toxicity is manageable, but long-term issues we're coming to learn about, not least of which secondary cancers. Other things include really very important recent data showing that the genetic signature of cancer that survives melphalan is much more adverse than if you don't get melphalan. So what we're hoping with melflufen is by being more targeted, taking advantage of stemness, but at the same time avoiding the side effect profile that melphalan has, that we will in many ways be able to overcome some of those challenges. My personal belief, Cindy, is that you need to use melflufen best in combination, because I think that's where we'll really get the maximal benefit of the drug. And we're working to figure out how best to make sure that it doesn't carry with it in the longer term some of the challenges that we see from melphalan.
So right now it's being used as a single agent or in combination with dexamethasone, or how is it being studied?
Right. So in the FDA approval will be for melflufen with dexamethasone. And we know that that's important. We know that dexamethasone matters even if you've had dexamethasone before and even if you've become resistant to dexamethasone, because that's actually part of the biology of the way it works within the cell. So we know that that's important. What's really exciting, and Darminder's data were the sort of first preclinically to show this, that when you combine it with other drugs, things go into a very different level of attacking the myeloma. And the really good news is that the combination studies led by my colleague Enrique Ocio, it's an international study group, the ANCHOR study, and Enrique's provided fine leadership with Mari-V Mateos as one of the senior investigators on that study. Basically, we've been able to show in the ANCHOR trial that when you combine it with other drugs, you know, things really go into a new level of activity. So the way to think of it is that it could become a very viable alternative to traditional chemotherapy because it would offer the promise of being more targeted and having fewer side effects.
Good. So if I'm understanding correctly, then I know one of the things that people are concerned about the other cytotoxic drugs is sometimes it causes damage to the DNA.
Yes.
Now, and that was a concern in the myeloma community about damage to the DNA with the stem cell transplant. The melflufen, since it's more directed, may not cause that damage. Is that what you're saying?
Well, we're hoping so. That obviously remains an area of important study, but I think it's very important to note that a lot of the drugs we give, be they chemo like melphalan and cytoxan, or even drugs like immunomodulatory drugs, can cause damage to DNA. They do. It's part of the challenge of exploiting the therapeutic index in cancer medicine. You have to deliver enough firepower to eliminate the tumor cell, but hopefully minimize surrounding damage to other cells. So our hope in melflufen is by having a more targeted approach, that therapeutic index will be better. In other words, we'll be able to spare potentially some of the side effects that we see more typically from other chemotherapeutics. But I would emphasize this is an area of active study. And we've clearly shown from our studies that myelosuppression exists with melflufen. And we also know from our experiences with prior chemotherapies that prolonged exposure to these drugs over time may enhance the risk of secondary leukemia, for example. With all of that in mind, that's why I'm particularly personally excited about melflufen being combined with other drugs. I could foresee, for example, where we would use melflufen, bortezomib, and then the CD38 antibody, achieve a fantastic response. It's just outpatient therapy, no hospitalizations needed. And then you would then go into a maintenance phase with drugs that we know are less associated with what you're just describing. So you can imagine how we would use this intelligently to exploit what I call therapeutic index.
Great. And something you said earlier that caught my attention, that you saw some activity with melflufen in extramedullary disease. More so than other types of drugs that we?
Yes, that's an excellent question. And Cindy, I appreciate you emphasizing it. So extramedullary disease is a challenge for us. This is where the myeloma has escaped the bone marrow and is sitting outside of the bone marrow in sites like the liver, the lymph nodes, the pulmonary area, be it within the lung or outside of the lung. These are very challenging manifestations of myeloma spread. And what we've come to realize is that all our drugs work against it initially. The problem is that over time, resistance characterized by escape in the extramedullary compartment is becoming a real problem. And we're seeing this particularly after, say, antibody treatment has failed. So what we were very impressed by in our study was that we were enriched for patients in whom antibody treatment had failed them and they had extramedullary disease. And obviously, if you have extramedullary disease in the first place and you receive an antibody, the response rate's around 20%. But once an antibody has actually failed you and you have extramedullary disease, obviously that represents an even more difficult situation. Remarkably, in that population, we're seeing about 25% of patients responding to just melflufen and dex. Why I'm so excited about that is I think if we simply add a proteasome inhibitor or add other drugs to that, we could see this go up a notch. So we now have a real potential answer to the challenge of extramedullary disease.
That's great. Wonderful to hear. And one last question before Yalak. Does it cross the blood-brain barrier? Have you seen it in leptomeningeal myeloma?
Cindy,
that's another unmet area.
Such a great question. I'm so grateful for you raising that. No, we would not expect that based on the biochemistry, but we have other drugs that do cross the blood-brain barrier. Selinexor is one of them. We know that some of the immunomodulatory drugs like pomalidomide can do it. And there's a great proteasome inhibitor that in my mind warrants much better study called marizumab, which we know crosses the blood-brain barrier. In fact, it was tested in pediatric brain tumors and in glioblastoma. Interestingly, in pediatric tumors, it's active. In glioblastoma, the phase 2 studies were very interesting. The large phase 3 that was launched has been fully enrolled. And my understanding from it is that just like a lot of glioblastoma trials, the results are not very clear cut. And that we're not entirely surprised because remember, glioblastoma is a very different set of diseases to myeloma. But we're on the cusp of launching a study led by my colleague, Dr. Clifton Moe, of marizumab with pomalidomide in CNS myeloma to address your point. But I see promise for CNS myeloma by virtue of combination strategies like selinexor, for example, hopefully marizumab if we can get that across the goal line, and other approaches like it. But melflufen is probably not, it might be part of an approach that we would use in a CNS affected patient, but specifically crossing the blood-brain barrier, we would not expect.
Is marizumab targeting CD38 too, like the others?
No, it's actually a very interesting drug, Cindy. Marizumab is a proteasome inhibitor. It's the most powerful proteasome inhibitor we have. It's more powerful than carfilzomib, and it's more powerful than bortezomib, and more powerful than ixazomib. It's derived actually originally from a marine source. And what's so interesting about it is it targets what's called the beta-5 subunit of the proteasome, but also beta-1 and beta-2. These are the mechanisms by which proteasome inhibitor resistance occurs. And we've actually done wonderful preclinical studies. Again, Dharminder Chauhan, my colleague, has been on the forefront of that. And then at the same time, we did clinical trials. And I'm really hoping the drug will continue to have momentum because for a variety of reasons, not really scientific necessarily, it hasn't seen as much study, in my opinion, as it should have done. And I think part of that was, sorry, Gary?
Is that the one that's been approved for use for myeloma in Australia?
No. The one that's approved for us in Australia is very interesting. That's also from a C source, from a C squirt. That's called Ablidin. And the Ablidin, we've also studied. Konstantin Mitsiadis at our center did the preclinical work, and we did a phase 2 trial with Dr. Mary V. Mateus. The reason it's not approved in the US is because the sponsor at the time of generating the phase 3 studies didn't engage with the FDA particularly well and had randomized trials where Ablidin was being given with dexamethasone compared to dexamethasone alone. And obviously in the US, we said, folks, no, that doesn't make sense. So to me, it's paradoxical that Australia has it approved because the Australians are very they have a regulatory apparatus with the greatest of respect. You know, I thank God for our FDA is all I would say, is that basically that's why it's approved idiosyncratically in Australia because you know, it worked, but no surprise because dexamethasone we know now is a drug from 20 years ago. It's not now. But nonetheless, I don't think it diminishes in any way the promise of Ablidin. And I rather hope that drug too gets its day in the sun because it clearly has effects, but it's not a proteasome inhibitor. Marizumab is a proteasome inhibitor, and it's what's called a beta-lactone proteasome inhibitor. And it's, in my opinion, one that really does warrant further study because I think, honestly, people thought with ixazomib, with bortezomib, with carfilzomib, we don't need any more proteasome inhibitors. Well, we all know now that that's not true. You know, ixazomib is a lovely drug, but it's not as powerful as carfilzomib, and bortezomib obviously is a mainstay. But, you know, as our patients live 15 and 20 years, we need the PIs later as well as earlier, right? And so I'm very hopeful that marizumab will continue. We're going to do our best to try and bring it to patients.
Thank you so much. And Yelak?
Yes. Hi, Dr. Richardson.
Yelak, a pleasure.
Melflufen is given the orphan drug and priority review designation by the FDA, right? So I want to double click on that. And the new drug application review is expected, I believe, still the end of this month?
Correct,
or has
yes.
Been pushed? What happens during that meeting? I want for patients to understand the mechanics of the approval.
No. My understanding, there's no ODAC. There's no. This is going through the traditional regulatory process that the FDA apply for an accelerated approval, and the date of declaration is supposed to be by the end of this month. My expectation is that we'll have that approval at any time because, again, in great credit to the FDA. You know, that they obviously understand cytotoxic drugs. There's no, when drugs have challenging and different toxicities, and the best example of that is belantamab mafodotin with the eye issues, which are very real, but fortunately we can manage them. You know, the FDA are obliged, and rightly so, to sort of say, well, wait a second, let's try and figure this out. And Gary alluded to it when we had with selinexor some real challenges from the GI standpoint for side effects. The FDA said, guys, we want to hear about this so we can really go through process to be very sure that the accelerated approval is warranted. And in great credit to that process, we then had the Boston trial where, you know, clearly selinexor performed extremely well against the control arm. And hence, you know, the approval, the full approval. So I think that you're going to see at the end of this month, hopefully, from the FDA, a green light to say melflufen is there for you as an accelerated approval. We then have a very important study called the OCEAN study. So Horizon is the study that's informed the accelerated approval. OCEAN is the study led by my colleague, Dr. Pieter Sonneveld, who's a very prominent leader in myeloma research in Europe. And he's actually the president of the European Hematology Association to boot, and a really great person. And he's led this phenomenal trial of comparing melflufen to pomalidomide and dex. And we will hopefully have a readout from that study fairly soon. And if that comes through, say later this year or next year, perhaps probably more likely next year to be fair, then we will be in a position to get full approval for melflufen in the same way as selinexor got full approval in December.
Nice. Okay. One of the challenges that you mentioned in previous meetings was what to do for patients that end up progressing on CD38.
Yes.
How do you see melflufen fitting in that algorithm? If I come to you as a patient and present myself, what is the mental algorithm you go through to say melflufen may be for you?
An excellent question. I would simply say that the CD38 failure in a patient, in other words, when a daratumumab fails a patient, or say isatuximab does, this is a major challenge for celiac, and I think that this drug class adds to our ability to meet that challenge, with additional combination approaches. So I think we'll be in a position to meet that challenge with these additional drugs. So, for example, melflufen in combination with something might be an ideal combination to do that.
Nice. Okay.
Great. So, my friends, I must apologize, but I have to sign off because I've just been paged. But it's been an absolute pleasure to work with you. And it's a great pleasure to see everyone. And I just want to wish you well and just say thank you so much for your interest in melflufen. And we greatly look forward to continuing our dialogue as we bring exciting new advances this year as well as in the future. So Priya, thank you so much, and I apologize. I've just been paged urgently, so I must jump off if you don't mind.
Thank you. Thank you.
Guys, God bless. Take care.
Thank you.
Bye.
Bye. Thank you.
All that, I think he covered a lot of information in such a short time.
Yes.
So I think this is definitely a talk I need to listen to again from the very beginning. So thanks for joining in, and Cindy, Gary, and Nalak, thanks so much for your questions. Hope we have a lot of listeners actually on this talk now. So thanks everybody for listening in. This talk will be available on CureTalk's website and along with its transcript. So thanks everyone. Stay tuned for more discussions on curetalks.com and stay safe. Thank you.